Welcome to Jerry's Gene Scene. In this video, let's look at the concept of codominance and incomplete dominance. Recall from our previous look at phantasma eye color that there are two possible phenotypes, and the monohybrid F1 cross resulted in a 3-to-1 ratio. One allele in this example, yellow eye, was dominant over white eye. We explained this by assuming that if the product to turn the eyes yellow was made on one chromosome, it was enough to turn the eyes yellow. Now let's look at the color of the spots on the body of our phantasma. There are 3 phenotypes. Can we use Mendelian algorithms to explain these 3 phenotypes? We find that blue spotted are pure breeding. A cross between blue spotted will always result in blue spots. The gametes are both blue spotted, and the resulting offspring of the cross is blue spotted, pure breeding. Yellow spotted are also true breeding. Forming the gametes, we can see that they are both yellow, and the cross represents the yellow phenotype. And a cross of 2 green-spotted phenotype results in 1/4 blue, 1/2 green, and 1/4 yellow. We know that both blue and yellow spotted are pure breeding. We can assume them to be homozygous. For the next step, I'm going to use the letter S to represent the gene for spots and label the allele for blue as S superscript B. And I will label the yellow as S superscript Y. Different texts and teachers may prefer a different approach, and as long as you operationally define what you want and are consistent, you will have no problems. So the genotype of our homozygous blue will be SB, SB, and of our yellow will be S superscript Y, S superscript Y. Can the hybrid cross of the blue and yellow produce a green? So let's take a look and see. And if we cross the blue with the yellow, we get green. So clearly, then, this one must be the heterozygous, and that would be SB, SY. So if we set up our usual phenotypes, genotypes, gametes, and offspring, we've got green, and we're going to cross that with green. And we've already established that that would be S superscript B, S superscript Y. In other words, it is heterozygous. So the possible gametes are SB and SY in both cases. And just like when we did the monohybrid cross, we'll set up the Punnett Square here, and we've got SB and SY as the two possible gametes, SB and SY as the two possible gametes. So we got SB, 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 SY, SB, SY, and SY, SY. So that's one that's blue, one that's yellow, and two possible green. So that's a 1 to 2 to 1 ratio. And so our prediction of 1/4 blue, 1/4 yellow, and 1/2 green is predicted using Mendelian genetic algorithm. Incomplete dominance and codominance are both types of inheritance where one allele isn't completely dominant over the other. The terms incomplete dominance and codominance are used interchangeably sometimes, and for the purpose of using Mendelian algorithms, there is no difference in setting up the gametes, the Punnett square, or the offspring ratios. The difference between the two terms becomes obvious only when looking at how the genes are expressed. In short, if the hybrid phenotype is a blend of the two pure breeding phenotypes, then it is probably incomplete dominance. Whereas if both alleles are expressed simultaneously, we would call that codominance. Thanks for watching. Please take the time to give this video a thumbs up and hit the subscribe and notification bell to get notified of future Gene Scene videos. And to learn to distinguish between homozygous and heterozygous, click on the video to the right. I'm Jerry Ward. I'm at genomealberta.ca.